Hey guys, so last week we had a lecture on the cytokine storm and how it happens. So I'll just simplify everything that was taught in that lecture. So let's start with what we know. A pathogen. Pathogens don't just come bald, like they don't just come empty. They always have baggage that's attached to them, and that is their downfall. So these are called pathogen associated membrane proteins. Okay, and a lot of different types of cells in the body, immune and non-immune, um, recognize these PAMPs, or pathogen associated membrane proteins, using pattern recognition receptors. Okay, and the most common ones are TLRs. Okay, um, like the most common types of PRRs. Okay, and so these cells will recognize this pathogen. And now this pathogen is not just going to sit and do nothing. It's going to damage the cells around it and kill the cells that it is able to. And so when these cells die, they release or they vomit out everything that they contain, including mitochondrial DNA, histones, S100 proteins. So all of these different types of proteins that are supposed to be intracellular are released outside. And so these are called DAMPs, or damage associated membrane proteins. Okay, and these DAMPs are going to go and complain to the macrophages. Okay, so these DAMPs are also able to bind to their own TLRs, their own like pattern recognition receptors, and so that will activate this macrophage or this dendritic cell or this epithelial cell. Okay, and so what happens when this macrophage is activated? So here you have the bacteria, and in blue you have the macrophage. Okay, so there's a connection between the PAMP and the TLR, and that is going to activate a pathway, like different pathways. Okay, and so to simplify, there's two main pathways. Okay, there's NF-kappa B pathway, and then there's IRF. Okay, and these pathways go to the DNA and order the DNA to transcribe and then later translate them into these proteins. Okay, however, transcription and translation takes a long time. This is a long pathway. Like if your house was on fire, you wouldn't go to the shop and buy a fire extinguisher. That would take too long. So there has to be a faster way. You have to have a fire extinguisher at home. And so these cells already have pro IL-1 beta or pro cytokines. Okay, so basically inactivated versions of these cytokines that are ready to be activated if a bacteria or any pathogen comes and activates this thing. Okay, so how do these IL-1 beta, or how do these cytokines that are already formed, how do they get activated? So there's this thing called an inflammasome. Okay, so it looks like this from above, and it looks like this from a sagittal view. Okay, and it contains these pieces. Okay, so this is one piece, and now this green thing or this dark blue thing here, okay, this is called a sensor. Okay, so this sensor is going to recognize the PAMP, or the presence of a PAMP. Okay, I don't, it's like a very complicated pathway that's still under research, but basically there's a sensor and it senses it. Okay, and when it senses it, so here, like it senses it, it activates this dark blue part, which is called caspase 1. Okay, and caspase 1, what it's going to do is it's going to activate this pro cytokine. Okay, and so you're going to have active IL-1 beta. Okay, so now what does this IL-1 beta do? Okay, so this is the short pathway. Okay, so what does this IL-1 beta do? First of all, the macrophage that releases this IL-1 beta, this IL-1 beta goes and binds to the IL-1 receptor on the same cell, and that is going to induce more IL-1 to be released from the same macrophage, and this is called autocrine action, okay? Next, it's going to go to the nearby endothelial cells, okay, so near to the macrophage, and it's going to stimulate the release or the expression of E-selectins, ICAMs, and VCAMs, among other things, okay? And so these are going to be expressed. And so this, and also it's going to stimulate the production of nitric oxide and prostaglandin E2, and this is going to make it's going to lead to vasodilation in the blood vessel, and it's going to make this the barrier more leaky, okay? So it's going to make the blood vessel more leaky. And so this is called paracrine action, okay? And then also IL-1 beta will stimulate these endothelial cells to release other cytokines other cytokines, and these cytokines will go and call all the other immune cells, okay? So like the neutrophils, macrophages, lymphocytes, okay? So all the other immune cells to come to the site as well. And this is called endocrine action, okay? So now that we have IL-1 beta out of the way, we can talk about the other two main ones. So there's TNF alpha and there's IL-1, IL-6. So mostly these three do very similar actions, okay? Like endothelial activation, just like how we explained for IL-1 beta, it promotes production of these same cytokines, so IL-6 and TNF alpha and IL-1 beta, and it induces apoptosis, so like programmed cell death in like certain cells that have a lot of that are damaged. And the most important thing is it causes or it stimulates dendritic cells to express more MHC1 and MHC2, okay? And when it expresses more MHC1 and MHC2, it's going to be able to show more of the pathogen peptides on its cell. And so when the dendritic cell goes to the lymph node, it's going to make it easier for the lymphocytes there to recognize the pathogen peptides and get activated, okay? So another paracrine thing that IL-6 can do is that it helps lymphocytes in survival and differentiation. And specifically, IL-6 are very good at promoting the T cell to differentiate into T follicular and T helper 17 like lymphocytes, okay? So basically activating the adaptive immune system, okay, both of them. Now, like the endocrine side of it is they both induce fever, and we'll explain how, and they both stimulate the release of acute phase reactants, like what? Like C-reactive protein, which is released by the liver. And C-reactive protein is very good at sticking to the pathogen so that it can be marked for phagocytosis, okay? So now we'll talk about how these three cause fever, okay? So first of all, they go to the hypothalamus, which is in the brain, okay? And what is it going to do? So here you have the blood vessels that are near the hypothalamus, okay? So these are the endothelial cells of the blood vessel. And this is the preoptic nucleus. I just colored it in. So this is the preoptic nucleus, okay? So TNF alpha and IL-1 beta, they're very big molecules, okay? So they're going to stay in the endothelial cells. They're only going to act on the endothelial cells directly, okay? And IL-6 is a very tiny molecule, so it's able to go straight into the preoptic nucleus. But their function is the same. So location is different, but their function is the same. What is their function? What is their job? They are there to increase COX-2, like stimulate the function of COX-2. COX-2 is an enzyme that converts arachidonic acid to prostaglandin E2, okay? And what is prostaglandin E2 going to do? It's going to go into the preoptic nucleus and tell the preoptic nucleus look, we need the temperature set point to be increased, okay? So can you please increase the temperature set point? So when the temperature set point increases, now the whole body is trying to increase the temperature, its temperature. So what is it gonna do? First of all, it's gonna shiver, okay? So the muscles are gonna shiver. Why? Because when you move something, it increases in temperature, okay? So shivering is one thing, and peripheral vasoconstriction. So all the peripheral blood vessels, like the blood vessels of the skin, all of those vessels are going to constrict so that all the blood can go to the 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 middle of the body, or where the infection is, okay? And so I call this central vasodilation, okay? Because middle and then vasodilation. And also the vasodilation, remember we said that there's vasodilation because of these cells. They stimulate the endothelial cells in the blood vessels to vasodilate by producing nitric oxide. Okay? So there's more blood going towards the site of infection, and these cytokines are causing vasodilation. Okay, so a lot of the blood is going to go to the middle. Okay, and now that there's a lot of blood, the neutrophils that were called are also going to come. Okay, and we said that there's more E-selectins that are expressed, more ICAMs that are expressed because of the cytokines, because of the three main cytokines. Okay, and so more neutrophils are going to come, and they're going to do diapedesis. They're going to enter the tissue. So what do they do when they enter the tissue? They're going to see the bacteria, okay? And now the bacteria is opsonized or attached or marked by lots of different things, like complement 3 C3b, C-reactive protein, which was from the liver, the IgGs, and also its own PAMPs, okay, so its own pathogen associated membrane proteins. The neutrophils are going to recognize it, and what is it going to do? It's going to phagocytose it. And when it phagocytoses it, it has to have a phagosome and lysosome, and then those fuse, and the lysosomes have a lot of reactive oxygen species, okay? And these reactive oxygen species are going to be released out, okay? And at the same time, these neutrophils are going to release elastases and myeloperoxidases, and these cause tissue damage, okay? And what do we get when there's tissue damage? DAMPs, okay? Also, these neutrophils release nets. And what are nets? They're basically so neutrophil extracellular traps, okay? Basically they're traps. And what are they made up of? Essentially DNA and histones, okay? So these, like DNA and histones, they're also DAMPs, like they're basically DAMPs, okay? And what do DAMPs do? They're gonna go and complain to the macrophage, and or any cell that's gonna listen to them, like any cell that has pathogen like PRRs are gonna recognize them and do something about it. What will they do? They're gonna release even more IL-1 beta, even more TNF alpha, even more IL-6. And neutrophils directly release these cytokines, okay? Now, this is fine as long as it goes down after a while, and as long as there is some kind of regulation. But when they're being released left and right from everywhere, from macrophages, neutrophils, like every cell is producing them, they spill over into the system, okay? So now these cytokines are not just affecting the infection, the area of infection. They're going to be everywhere now, and that's going to cause vasodilation everywhere. And that's not good, because it can compromise your vitals, your vital signs, okay? So how exactly is it going to do that? Like, I'm going to explain like one of the most dangerous things that can happen, okay? So IL-1 beta and TNF alpha, they're going to go to the liver, and they're going to cause it to release protein C, antithrombin 3, things like that, okay? So these proteins. And IL-6 goes to the endothelial layer or endothelial cells and cause it to release tissue factor, okay? And there's going to be damage. So we know that because of the infection and all of that, there's going to be damage to the endothelial cells. And when there's damage, it's going to expose the subendothelial collagen. And IL-1 beta and TNF alpha, they both activate platelets, okay? And NETs also activate platelets. Now, what is going to happen when all of these factors are present? You're going to increase the coagulability, okay? So when the and there's vasodilation. So what happens when there's high coagulability? The blood will clot. And when it clots, what can happen? What's the most dangerous thing that can happen? DIC. Okay, so what is DIC? Basically, because of the high coagulability, the blood is going to clot in a specific place. When it clots in a specific place, other blood vessels are not going to have blood that goes to them, okay? So that's why other places will not have that many red blood cells. And so it's going to lead to hypoxia, and those organs that don't have blood going to them can fail, okay? And that's fatal, okay? So yes, acute phase reactants are good. I mean, these like first order cytokines, they're good, they're helpful, but to a degree. Like you can't have so many to the point where it'll destroy not just the pathogen but yourself as well. Okay? And so there are certain medications that can fix this, like if there is like a cytokine storm. So how to fix it? Anti-IL-1s are like anakinra, TNF alpha, infliximab, IL-6, tocilizumab. The way I remember IL-1 is like one is like ana al-awwal or like ana, I don't know, okay? So one, like I'm first or whatever, okay? Anakinra. And then glucocorticoids, obviously, they basically dampen the whole immune system. Yani they suppress the whole immune system, okay? And then TNF alpha, IL-6, okay? So yeah, basically you can fix them with this, okay? And that's it. So just wanted to thank Dr. Farhan and Dr. Amara for teaching us. We really appreciate you guys, and then Janeway for the pictures and things, and of course we can't forget ChatGPT for making life incredibly easy. Thank you.